So very happy to be here with you and to discuss this very important topic about the roles of neutrophils and more specifically of neutrophil extracellular traps in sepsis. So NETs are really casting a new light on sepsis management. Bienvenue, as we said in French, again. And so what we will be learning today is that neutrophil extracellular traps are produced by ejecting chromosomal material out of a cell. They catch and kill the bacteria and viruses. They can sterilize the blood in minutes, so very fast action. So a long time we know about NETs that have been reported first in 2004, so 20 years ago. And there is more than 5,000 publication in this topic in general, but also more specifically in sepsis. So sepsis, as you know, is really putting a great pressure worldwide. The burden of sepsis is highlighted by the numbers you can see here. Almost 50 million people are affected by sepsis on a yearly basis, according to WHO. 11 million people are dying from sepsis on a yearly basis. Just to let you know, in Europe is about close to 700,000 deaths yearly related to sepsis. Many children are suffering from sepsis, so it's really a huge problem, a huge burden, social, economic burden on our societies. It's the number one cause of death in hospitals. It's also a major cause of hospitalization. So about 1 out of 3 patients who survive from sepsis is going to have recurrent sepsis in the following year and be readmitted to the hospital. And the related cost is also tremendous. It's more estimated to be more than $60 billion in the United States alone. And unfortunately, death is not the only price to pay. And if you survive from sepsis, unfortunately, you are at higher risk of having long-term sequels, cognitive dysfunction, but also motor disabilities, and in some patients, an increased risk of new onset of new disease, could be chronic inflammatory processes or even autoimmune diseases. So again, I would like to really thank Volition for organizing here that symposium to help us learning more about NETs and their role in sepsis. So Volition has developed a low-cost, user-friendly, rapid diagnostic test that may allow detecting patients who have high activity of NETs that may be driven the sepsis in the patients, and that therefore may help interacting with patients' trajectory toward improving patients' outcome. So we are here to present the new QNETs H3.1 assay, so measuring the circulating levels of H3.1, which is one part of the whole NETs. It's a novel and clinically relevant biomarker which has a potential to change the management of patients with sepsis. So to start with this satellite symposium, so we have, and you will see in the subsequent talks, that we have data coming from 3 independent large studies for more than 3,000 patients with consistent findings across these independent cohorts that help clarifying that elevated H3.1 level reflects the deregulated immune response and is associated with an increased risk of death, an increased risk of progression to septic shock, an increased risk of progression to multiple organ failure, an increased risk of progression to acute lung injury and ARDS, as well as an increased risk in progression to acute kidney injury related to sepsis. And also, and this is the very next future, it is very likely that measuring H3.1 could help severing like as a treatable threat in the sepsis management. So, as I mentioned, we have three outstanding speakers this afternoon, and I would like first to invite Andy Retter, who is a consultant in critical care, ECMO, and thrombosis, and a clinical leader for critical care at Guy's and St. Thomas hospitals, has done a lot of work in immunothrombosis, in the relationship between the immune response and the coagulation response, and of course has tremendous work in the field of nets. He's also the CMO for Volition.
Thank you very much for that introduction, Djillali. Thank you to everyone who's joined us here today. It's really exciting to see so many faces. And our job today is to take you through the H3.1 test. We're going to try and explain a little bit about the biology of netosis and why when it becomes disordered that can be a problem to our patients. Be delighted, my colleague Terry Kelly from our laboratory in California is going to speak too, and really sort of justify or prove the test is detecting what we say it is. And then Caroline and I are going to take you through some of the clinical data from those 3,000 patients that Djillali mentioned to you. The other point I want to get across is that this is a blood test that's going to be run on a purple top tube. It's going to be run on a K2 EDTA tube. You should do it in about 15 minutes, so it's no extra testing to your patients, and we can add it to sort of routine laboratory testing, and that's really exciting. It's not invasive and shouldn't be burdensome for your patients. Just need to say that I am an intensive care consultant. I'm also a hematology consultant with a particular interest in thrombosis, and I do work at Guy's and St Thomas's. I'm paid to be here today by Volition, and the opinions I'm expressing today and the data I'm sharing today doesn't reflect on my trust. It's from Volition. And just to say that I am paid, and I do have shares paid for by, I do own shares in Volition. This is what we're detecting. This antibody here is binding to the nucleosome and picking up the H3.1 histone tail, and it's very specific for those nucleosomes circulating in the blood. What's a nucleosome? Well, it's 147 base pairs of DNA wrapped around a histone optima, and those histones are really critical for packaging and storing our DNA. I think I've mentioned these bits to you already. It's a 15-minute test. It's done on a purple top tube and shouldn't involve significant extra testing for our patients. What's the normal range? Well, we think the normal range in healthy adult patients is less than 30 nanograms per mil, and we can easily detect down to 20 nanograms per mil. And the upper limit of quantification of linearity is maintained up to 20,000, and we'll show some data later as to why that's important. You can get nucleosomes into your blood in three ways. You can get them from normal apoptosis, senescence and death of cells. You can get it from tissue injury and necrosis. And most importantly, in sepsis, you can get it from neutrophil extracellular traps. And this sort of eruption, this sort of fungal hyphae ejecting from the back of the neutrophil, we know now in sepsis, and Terry will talk a little bit more about this data later, that over 80% of the nucleosomes in sepsis come from these neutrophil extracellular traps. They are, or they can cause badness. Simply put, they are very disruptive. You can see down here, they are directly toxic to endothelia. They disrupt the phospholipid bilayer, and they can cause inflammation when they're released to excess. They can also directly activate platelets. They can activate complement, and you get this sort of feed-forward loop, which leads to extra neutrophil recruitment, extra release of neutrophil extracellular traps, and this sort of dysregulated response that moves away, and you lose localization and lose control of it. Lastly, here we're picking up there is crosstalk and talk to other cells of the innate and then adaptive immune system through the toll-like receptors. Really, H3.1 sits at the triumvirate of innate immunity, inflammation and coagulation, and it's an excellent marker of activation of your innate immune system. and its pathology is due to its indiscriminate binding of the anionic components of the nucleosome with critical endothelial beds in the circulation and vasculature. If you look, this slide is adapted from Ella Silk's paper, which was published in 2017. Ella was a trainee with us. If you look hard enough, you can see their effect and pathology on every vascular bed. We're going to pick up here, and when we talk about our clinical data, how it particularly affects the lungs and how it particularly affects the kidneys. And perhaps that's the easiest place, or place where the signal is the strongest at the present with the data that we have in hand. This is a simple model of immunothrombosis, or perhaps not that simple. What do I mean by immunothrombosis? Well, immunothrombosis is the link between our immune system and our hemostatic system, which is activated. Our hemostatic system is activated when a patient is infected. What we're trying to show at the top here is an alveolar sac, invasion of a bacteria breaking through the alveolar capillary wall, and then you get recruitment of, or activation of complement, activation of neutrophils, activation of platelets, and they come together. You can see this ejection of chromatin behind the neutrophil, which acts as a scaffold. It's decorated with proteins which recruit other cells of the innate immune system and adaptive immune system. It can activate platelets, activates complement. You see here the alternative pathway or the intrinsic pathway of coagulation activated and how that can lead to extra complement generation. And you can see how our natural anticoagulants link into it here as well. I could talk for a very long time about immunothrombosis, and I'm going to pause there. I won't bore you or distract you further, but it's an incredibly localized process. It's only supposed to be occurring exquisitely at the site of tissue injury and tissue infection, and it's temporally extremely localized as well. It's only supposed to be occurring at the time that that injury is there. And one of our arguments is when you get disruption of that localization or those localization processes, that's when you see downstream pathology, and that's really critical. I'm going to hand over to my colleague Terry now, who's going to take you through some of the science of the test and actually proving that we're measuring what we say we're measuring. Thank you.
Right, thank you everyone, and thank you for the opportunity to come and speak with you today. So first here I wanted to draw your attention to the figure that's on the right. This is, as Professor Annene mentioned, this is actually a neutrophil in orange, and it's actually the chromatin that is being spread out. And what you can see there in the purple is this is where it's actually capturing the bacteria. So this is the main role of these neutrophils, is to remove these pathogens from circulation. But you can also see the beginnings of what Andy was talking about in the challenges. That's a red blood cell that's also being caught up in that net. And so in this one picture, you can see both the value of having these neutrophils and removing the pathogens, but also the potential danger if you don't control netosis properly. And on the left is just a schematic which shows the actual neutrophil releasing its chromatin contents, and in those nets is that chromatin core and the nucleosomes, which is very much at the heart of what we're studying with the Nu-Q assay. So we've gone about showing that really what we're measuring is nets in the laboratory, but also in patients as well in a variety of different ways. So here in our immunoassay, we use two different antibodies, one that recognizes the H3.1 histone and a second that recognizes the nucleosomes. So this is immunostaining here of isolated neutrophils that have been induced to form netosis. And so what you can see in the green is on the top is our H3.1 antibody, and on the bottom is our anti-nucleosome antibody. And you can see that these stain the cells and go along with the DNA along the nets. The next figure I'm going to show you on the right, this is actually an image from our colleague Dr. Indra-Aswani, and so here you can actually see the neutrophils ejecting their chromatin with nets. So this is using a DNA stain. And so I think first is just to really appreciate the size of these nets from these individual cells and how much space they can take up. So on the right, this is the DNA being released, but as we're measuring an immunoassay, what we've done in the lab is we've correlated the release of this DNA with our nucleosomes using our Nu-Q assay. So on the left is a graph where we're overlaying where we've isolated neutrophils, we've induced netosis, and in the triangles we're measuring the release of DNA, and we're correlating that with the Nu-Q levels. So it can show that in the lab the DNA release is correlated with nucleosome levels. As we transition into the clinic, it's important to know a couple of things about nucleosomes. So one is their release is just sort of normal processes, and in terms of cells are dying as every day. We've done a lot of studies that show that H3.1 levels are not impacted things like height, weight, age, and sex, and it's also not impacted by the circadian rhythm. And this is because while your body is normally releasing nucleosomes, there's also exquisite mechanisms to help clear them out from circulation. So really what we're measuring ultimately then with the NuQ assay is this imbalance in nucleosome release and clearance. So here I'm going to take you through some data that we've received through sequencing. And so first I'll start on the right-hand side. And so what this is a graph of looking at the length of cell-free DNA in circulation. So on the x-axis is the DNA length, and on the y-axis is density. So that's how much of the DNA is in these various size bins. And so as Volition studies both cancer and sepsis, what you'll see first with those cancer samples is that a majority of the DNA is small being less than 500 base pairs. So these tend to be in these mono and dinucleosome fragments. Whereas when you look at sepsis samples, you'll see longer chromatin fibers. This can be very much likened to the schematics on the left when you look at different ways in which DNA is released with cell death. So in chromatin decondensation that occurs with apoptosis, you get this unwinding of chromatin, leaving these linker regions accessible. You'll have enzymes that will come in and chop up these chromatin fibers into small mono and trinucleosomes. Whereas in NETs, the process of DNA release is much faster, and you'll get these much longer fibers. And so this is part of that explanation about why you'll get these longer chromatin fibers in sepsis compared to cancer. This is all still very much correlational, and so we wanted to look at how do we know that this DNA is really coming from neutrophils or through NETs. So to do this, we've taken advantage of some DNA methylation data. And so for those of you that don't know, you have different DNA methylation patterns in all of your cells. This is how you go from a single genome to all of the different types of cells. And so what we have taken advantage of some DNA methylation atlases that we can apply to cell-free DNA and essentially deconvolute the DNA to know where is the DNA coming from, what cell type, what tissue type is it coming from. So in these two examples here, we looked at our sepsis samples, and when we deconvolute the data, we can see a majority of the DNA in those sepsis patients is coming from neutrophils, compared to in this NHL sample, they were mostly coming from B cells. And this was good because this was actually a B cell lymphoma. So this was some of the early data that we had that supported that we could use this methylation-based cell of origin to identify where DNA was coming from. We continued this into a different population of non-cancer patients. And so here we have on the left is a group of healthy cohorts. In the middle are very sick patients that are in the ICU but are not septic, followed by our sepsis patients here. So again, on the x-axis is size and the length of DNA. Here, each line is an individual patient, and it's a heat map. So the more red it is, the more DNA, the more material is there. And so what I hope you can appreciate is that really these sepsis patients stand out. And so the DNA in these sepsis patients have very long fragments of chromatin that differ from what we see in the healthy and in the non-septic ICU patients. When we then applied our methylation cell of origin analysis, you can also appreciate there that in the sepsis patients, we have a much stronger contribution of DNA that is coming from neutrophils. Again, this is helping us correlate then that these elevated nucleosome levels we're seeing are actually derived from neutrophils and have that fragmentation pattern, which we would expect to be coming with NETs. And so with this, I'll turn it back over to Andy to share some of the recent clinical data.
Thank you, Terry. So, as I said, I wear two hats as a haematologist and as an intensivist. And certainly that data is really important to us because it underpins and is the methodology underpinning the validity of the test. And as we take it forward and start to work with regulatory partners, that's really important to show that. And that's more evidence than there are for things like CRP, for example, that we all use so routinely. This data was some of the data I'm going to show you now was previously presented at our key opinion leader meeting, which we held about a month ago. Djillali and a number of other experts were there, and we're very grateful for their opinions and help trying to shape this project and take it forward. I'm going to cover data from 3 cohorts, basically. We looked at just under 1,000 samples from the CIS-PCT study in Germany. That was with my colleague Caroline, who's on the panel with us here, and Professor Michael Bauer from Jena. We're very grateful to them. We've looked at about 1,700 samples in the Mars cohort and been very well supported. Very grateful for the support from our colleagues from UMC Amsterdam and then Djillali's own group in Paris. Another slightly boring slide. What does this show and why have I labelled this point? Well, H3.1 weakly correlates with your neutrophil count. Why is that important? Well, it's really important because H3.1 tells you something your neutrophil count doesn't do. You don't just have to measure someone's white cell count, it's showing you information over and above the white cell count. So that's a really critical thing and one of the first things we checked when we looked at the data. Then we scanned the data to see were there any signals of badness. We've presented you a model of immunothrombosis. I didn't really talk about thromboinflammation, but that's when that process gets dysregulated and you get distal damage to organs away from the site of infection. So could we see a mortality signal? And yes, we started to see a mortality signal. These are patients from the Jena data set. There's 1,000 patients or 971 patients in this. And you can see this figure here is just picking up the patients with a very high level, 43,000 is the sort of current record, died quickly. Tabulated, that's this. Every patient with an H3.1 level over 20,000 died. There was a 25% mortality if you had a level over 10,000, and you can see how it goes down. Plotting that on a Kaplan-Meier curve, you can see that going out to 90 days survival, we were able to distinguish a cut-off threshold of 1,140 nanograms per mil. So there was a clear impact and a statistically significant impact on your 90-day survival. And you can see the figure there, 3.1 times 10 to the minus 9. It's really important for me as a clinician, but also for Volition as a company to be open and transparent with our data. So all of our data has been analysed in the statistical package R, and we use the program in that called SERV-MINOR to show that. And so when we publish our data and when we share our data with regulators, they can review and see how we've actually done our homework, so to speak. I haven't put the mortality curve here, but if you look at 28-day mortality, that cut-off changes slightly to 2,600. But we're starting to produce really clinically relevant numbers that you could use. So normal is less than 30, over 1,000 is high, over 2,500 is very high and linked to pathology. And I'll come back to that in a second. I'm going to hand over to Caroline now, who's going to take you through some of the specific data relating to acute kidney injury. Thank you.
So I want to show you our data on the clinical utility of Nu-Qs in septic acute kidney injury, with which we try to find, or which would we try to improve the AKI classification, and with which we try to improve and target an early intervention in AKI. So I would like to thank the SAPNet Critical Care Trials Group for providing us the data, which really deliver a valuable insight into this complex condition. So please let me start giving you a brief summary of the pathophysiology of septic AKI, because as you know, it phenotypically differs to other AKI forms. It's really complex, and this slide may be explaining why single target therapies often have failed. So in this so-called unified theory of sepsis-induced AKI, Gomez and colleagues nicely could show that in sepsis-induced AKI we have inflammation and oxidative stress, which produce endothelial and microvascular dysfunction. And we have a decreased red blood cell velocity and increased transit time of activated leukocytes. And all these mechanisms together, they trigger and they increase local inflammation, which is leading to a regional hyperperfusion and hypoxia. And another main aspect is the adaptive bioenergetic cell response, which is a response of the mitochondria and leading towards cell survival. So in sepsis-induced AKI, we have a disrupted bioenergetics leading to a mitochondrial dysfunction and leading to a reduced energy production. So in sepsis, we have the release of PAMs and DAMs and other inflammatory mediators into the circulation, and they are getting access to the peritubular space through glomerular filtration. And once they are in the peritubular space, they can they can induce kidney damage at different molecular patterns. So, for example, LPS can directly interact with the toll-like receptors at the surface of the renal epithelial cells, or they might directly interact and stimulate the cells. And this is leading to a kind of paracrine signaling, which is inducing such a mitochondrial state in the tubules. And in the last years, there have been significant advances in the understanding of the role of immune cells in early injury and repair from AKI. So resident immune cells are present in the normal kidneys, and in kidney damage, they induce inflammation, which is followed by a rapid infiltration of these cells. And DAMs and PAMs, they enhance this immune cell recruitment, leading to or participating in renal damage, leading to apoptosis and necrosis of tubular cells. And in some patients, we have an adaptive repair with a tubular regeneration and a complete resolution of organ dysfunction. In other patients, we have such a maladaptive repair, leading to tubular loss and the progress of a chronic kidney disease. So, as we already have heard, activated neutrophils are doing netosis, releasing their cytotoxic content like DNA, histones, elastase, and of course they activate the complement, and they are part of the establishment of acute kidney injury. So we have a disbalance between the net production and the net degradation. So netting neutrophils are in circulation, they infiltrate the kidneys, then they induce a direct damage through releasing their cytotoxic content, and they have a lot of indirect damages like inducing thrombosis, exacerbating ischemia, and they perpetuate inflammation, which is going into a vicious cycle. So, with this pathophysiological background, we conducted a study, and now I'm coming to our clinical data. So we have done a secondary analysis of patients recruited for the CIS-PCT trial. So CIS-PCT was a multi-center trial evaluating the effect of high-dose selenite versus placebo and a PCT-guided therapy versus standard of care on mortality in patients with sepsis and septic shock. And in these patients, we evaluated levels of circulating H3.1 nucleosomes as a surrogate of netosis. And we tried to find an association between the development of AKI and its severity. So our primary endpoint was the development of AKI within 7 days. This was based on the current KDIGO guideline by an increase in serum creatinine from baseline. So of course we have to exclude patients who are on pre-existing renal replacement therapy. So 881 patients were eligible for final analysis. Of these patients, 7.6% they progressed to stage 1 AKI, 3.2% to stage 2 AKI, and nearly 25% of the cohort they progressed to stage 3 AKI, of whom 86% received renal replacement therapy within 7 days. And to have a comparator, we of course evaluated patients who did not develop AKI, so 64.4% of the complete cohort did not progress to AKI. So this is our measurement protocol, so to have a more dynamic process of the levels of NETs and their correlation to acute kidney injury. We measured at different time points, so day 0, randomization, day 2 and day 7. And of course, we correlated them to secondary outcomes like mortality or length of stay or renal replacement therapy dependence. So this is a short excerpt of our cohort. So the baseline characteristics show that the male gender was equally distributed within the whole cohort. And of course, what you can see and what you could expect, that patients with the severe AKI, they were significantly sicker than the other ones, as expressed perhaps in the SOFA score. They had more organ dysfunctions. They had higher levels of lactate and adrenomedulin, and 70% of them already were on septic shock within admission, and 80% of them already were invasive mechanically ventilated. So these are the patients who did not develop AKI, but who have a pre-existing renal impairment in the screening process, so 24 hours before randomization. And what you could see, the more pronounced the reduction in GFR, the higher the H3.1 levels at baseline, maybe due to an altered clearance or maybe due to a low-grade chronic inflammation of these patients. And in this slide, you could see the nucleosome levels over time within the whole cohort. So what you can see is that AKI severity correlates to H3.1 levels, so they are significantly higher in those having a severe AKI, with a peak at day 2 perhaps showing a more delay after initial injury. And what you see, especially in those patients who are on renal replacement therapy, we have a sharp decline in these H3.1 levels after inducing renal replacement therapy with to day 7. So we did further analysis. We stratified the whole cohort into quintiles, ensuring having the same population or the same size of patients, and allowing a better or fairer comparison of them. And this is the so-called creatinine quintile. So the x-axis are time in days, and the y-axis is the possibility of not receiving renal replacement therapy. And you see a consistent separation between the lines. And in the next slide, you could see the H3.1 baseline quintile, and you could see a more pronounced separation within these lines, especially in those patients having really high levels over 3,000. So in the final slide, you could see that we try to produce a clinical model in predicting renal replacement therapy within 28 days. So the components of our model was the H3.1 baseline level, which was over 2,600, the platelet count, which was below 80,000, and the urine output, 24-hour urine output, was below 13 ml per hour. And with this clinical model, we could predict the probability of RRT in 84%. So to summarize and to recapitulate my slides, so sepsis-induced AKI involves a complex pathophysiology, showing NETs playing a crucial role in this complex process. So H3.1 levels, they correlate with AKI severity, showing distinct temporal patterns, and particularly in severe AKI requiring renal replacement therapy. So a clinical model, which I could show you, incorporates H3.1, platelets, and urine output, who demonstrates a strong predictive performance for renal replacement therapy needing. So I hope I could convince you that these findings, they open up exciting possibilities for improving a personalized management of sepsis-induced acute kidney injury. Thank you.
Thank you. One of the problems with any biomarker is it correlated to, or is it causal? And so thank you, Caroline, for that talk. That was one of the things that was really picked up in the slide. So creatinine is inert and it builds up in kidney injury. The pattern is different here, and it's suggesting when you're breaching a threshold that actually these H3.1, these nucleosomes from NETs, are injurious. And that difference in the pattern there is quite a key thing just to pick up. We think that's where you're losing control, and immunothrombosis is evolving into thromboinflammation, and that's where you're getting tissue damage. We're quite excited by this model because many of you will use electronic healthcare records. We use Epic in my hospital. You could code this as an algorithm into it. It's just got 3 variables: an H3.1 level greater than 2,600, low platelet count and low urine output. excuse me, and that enables you to predict who may or may not require renal replacement therapy, so it's functionally very useful. We've worked with colleagues from a company called Santersis who have an immunoabsorption column which can remove H3.1, and that's been shown when it's been applied in an animal model of sepsis. It lowers the H3.1 level and has been shown to be protective of renal injury in a pig model of sepsis. Professor Tacconi and Professor Jean-Louis Vincent's laboratory have worked with a small molecule inhibitor of NETosis, something called MCBOS, and that was published in Critical Care last year. And that model was also protective of acute kidney injury in a sheep model of sepsis there. So we think that blocking and antagonising the action of H3.1 when it's excessive is a really interesting and potential therapeutic target for us. An immunoabsorption and a small molecule blocking it, that's two different mechanisms that we're using. In my main clinical practice, I deal with patients with respiratory failure and ECMO, and so I was particularly interested in this paper that was published recently by Mittendorf. The team in this study produced an artificial model of acute respiratory distress syndrome by instilling gastric acid into the lungs of pigs, basically. This slide just shows that they were good at creating ARDS. In that you can see the classic picture and infiltration on this H&E stain here, and you can see the chest x-ray changes and the lung injury score and the PF ratio moving in the directions that you would expect. We've worked a lot with these colleagues, and you can see that H3.1 has increased in this population. Again, you can see neutrophil deposition, you can see fibrin deposition here as well, and you can see another H3 marker is increased as well. So you're starting to see this as an animal model where NETosis is important and playing a key part in the pathophysiology of the disease. They used that column that I mentioned to you before, the immunoabsorption column, to reduce the level of H3.1 in those organs, and in the treated animals you saw less infiltration and less fibrin deposition. That's actually been optimised for lung transplant as an extracorporeal circuit for that. But that's a really interesting animal model of biology of NETosis causing organ damage, and actually you can treat it and ameliorate it in some way. It was only published about 3 weeks ago, that paper. I've known about it for a little while, but it's available online and the references are on the slides you'll be able to see. So we were really interested in our data. Is there a signal that we can see in ARDS? And yes, there is. So in patients who are defined by the sepsis 3 criteria, where they had sepsis or septic shock and had severe ARDS, as defined by their PF ratio, you could see the highest H3.1 levels. It's indicative and correlates with the severity of illness. So that's really quite exciting. You've got potentially a new test which can help predict the severity of ARDS and the patient's trajectory. And you've got studies suggesting it's something we could potentially treat or ameliorate to potentially change outcomes. Now that's a few steps away, but it's really exciting. And it's exciting, it fits with that immunothrombosis, vascular inflammation biology that I've been suggesting to you. We have an absolute ton of data now. We've tested H3.1 in over 14,500 samples, looked at over 3,000 patients. So I'm going to quickly try and rattle you through some of the other data, and I'm very, very, very happy to take questions on it. I can't present it all. Working with our colleagues in Amsterdam, there was a similar correlation as demonstrated in the German data with mortality, and that was particularly prominent on day 2. The higher the H3.1 level, the greater the risk of mortality is shown here. There was a similar relationship with acute kidney injury. The higher the H3.1 level, the greater risk of acute kidney injury. Actually, this is quite a complicated Technicolor slide, but picking it up here is that you can see the higher the H3.1 level, the greater the risk of multiple organ failure. And that's really going back to sort of Ella's slide that I mentioned to you before, where we're picking up damage in multiple vascular beds. Again, this is just a consistent signal across a completely different clinical data set where we've tested the results. This is work from our colleagues in Paris, and again, it's showing a repeat signal. Those patients who died had a higher median H3.1 level just the signal towards badness, saying it's a relevant marker to track. It looks like there is discriminative ability between sepsis and septic shock. In patients who are defined as having septic shock as per the sepsis 3 criteria, again, you can see a higher median H3.1 level of just over 1,000 is what we're picking up here. So that's clinically useful and tracks with severity of disease. Understandably, there's crossover between sepsis and septic shock. There's ever so slightly subjective on how we quite tease those apart and delineate them. This is a similar signal in that data looking at acute kidney injury. Again, the higher the H3.1 level, the greater the risk of acute kidney injury and requiring renal replacement therapy. This is a slide suggesting a hazard ratio, expect in a slightly different way, and it's looking at time of risk of requiring renal replacement therapy. And what you can see is that patients who have an H3.1 level greater than 3,000 in this data set, so very close correlation to the 2,600 I mentioned to you before, have an almost 12 times greater risk of requiring renal replacement therapy. And again, that's statistically significant, as you might expect. Again, it's a slightly smaller data set, this one, and the test was, and what you can see here is that if patients required invasive mechanical ventilation, they had a higher mean H3.1 level. Slight challenge with this data is that a significant proportion of the patients were ventilated, many more were ventilated than not ventilated, and they were sick patients. Their SOFA scores were 8, 9, so it's not that discriminatory there. But again, it's consistent with the other data that I've shown to you. I'll hand over to Djillali in a second, but just to recap, we've shown you some of the biology behind mitosis. We've shown you that the test actually measures what it says it's testing. It's in a purple top tube, done on the same blood test. You run a full blood count or complete blood count, doesn't require any extra testing, and we can turn it around in 15 minutes. And there is now a consistent signal across 3 large data sets of an injury to, or correlation with mortality, renal injury, and respiratory injury and failure. Thank you.
Wow. So we indeed had a fantastic talk this afternoon. And here are what I picked up from this. So I think what's very important is that the accumulation of data on a large number of patients with sepsis and the accumulation of data coming from different countries, different teams, acting independently and even not knowing that others were looking at the same things in the same time. So this is very reassuring. The consistency of the findings is very important, again, in a large number of patients managed in 3 different European countries. So I think that the evidence from the data that has been shown is that there is something related to high level of circulating A3.1 in patients with sepsis. It is clearly associated with a bad trajectory for patients. That is an increased risk of dying and also an increased risk of organ dysfunction. It looks like that precedes the development of organ dysfunction in patients with severe infection, and in particular at the kidney level. So I think this is very interesting, and in the next 15 minutes or so, it is very important that we discuss together the data that we have seen and what is the meaning behind this data and what are the steps to move forward in the very next future. So first of all, I would like you to join me in congratulating our three speakers today.